the key here, so just we create a trace file, and ultimately there's actually a three-step process, and I'll talk you through what we call our pipeline. But what you're getting is you're getting pulses from these nucleotides as they're getting incorporated. Those go through a camera, and then those basically are creating images, but that's happening across, in our case, we run 4 different wavelengths, 4 different cameras, so we have to reassemble all that, put it all back together, and that's where we just get the pulses. Then we have to take the pulses and look at it on a time domain, and the widths and the heights, and then we have to take statistical calls on whether or not we think a base exists. And that's the complexity on the algorithm side. I do touch base on that one a little bit too. Let me go back to DNA, kind of get you back to, okay, so you always have to kind of reset, what am I doing and where am I going with all this? So first off, you're trying to basically sequence DNA. You want to know what the order is. You're going to study a genome or a bacteria or some segment of a chromosome that you want to target. So you have to go through the process of extracting the DNA, and some of you may have done this. My daughter just did this in her high school stuff. In this case, these DNA extraction solutions, they're open on the market. You can get it for human genome. You can get it for different plants and things. There's different types of extraction solutions. At some form or another, you're going to be breaking down the cells. Human cells, you typically lyse them, and there's certain, there's just standard methods in the laboratory to lyse cells. And in the process, the DNA will typically clump together. You can extract it out and purify it so that you've got nothing but the DNA. That's the starting point for where we go. And so this is typically, like people that want to run our instrument, somebody's sending them in, here's some DNA, tell me what it is. And that's usually where it starts. In our case, what you end up with at the very beginning is these really long strands of DNA. And these can be millions of base pairs long. And that's too much for us to be able to handle inside of the SMART cells. So the first step, what we have to do, and all of this is occurring on a wet bench, next to the instrument, but you have to do this in advance of being able to start the sequencing. So we'll take that long fragment of DNA, and normally what we'll do is just a basic sonication to fragment it up. And after you fragment it, what you end up with is the smaller fragments. Actually, our target length is about 10,000 to 20,000 base pairs long. That's what we recommend right now with our current chemistries. But the fragmentation process will usually leave strands on the end. It'll leave nicks and gaps within the DNA. So you have to go through a repair process. So you repair the ends, and you clean up the insides, and there's just little enzymes that exist that we drop in there, and they repair the DNA. Then the next step we go through in the preparation process is we ligate these adapters on them. These are custom-made adapters because they have a specific purpose for us. And we put these adapters on the DNA fragments. Then we go through one more purification step because we want to make sure that there's no free adapters floating around. We want to make sure any DNA that didn't get its adapters stuck on the ends. We've got to get rid of all that. All we want is the DNA with its adapters on the end, because then we go through a step where we anneal this primer. This primer sitting inside of these adapters is where we anchor the enzyme, and that's this enzyme down here at the bottom. So this gets us to the point where we're ready to put this in the instrument and actually start the sequencing run. So here's our custom enzyme here, ready to go through and do the sequencing. I'm going to show you a movie of how this works. It's kind of out of place, but it's the only way I can show you. So think in terms of the movie I showed you before. The enzyme is stuck at the bottom of the zero-mode waveguide, and it's attached there hanging onto this fragment of DNA, and it's got to go through this process of sequencing. So what it's going to do is it's going to first unzip the fragment. So it's only going to work on one end. Then it goes around, and now it can start incorporating the base pairs. So now it's incorporating them, and from that point on, it's going to cleave off that strand and just keep going. And it'll go around and around as long as you're running the movie. If your fragments are really long, it may only do a single pass along the fragment. If the fragments are really short, it can do multiple passes and give you many X coverage. The more times around you go, the more accurate your DNA calls are. And that's how that works. So this gets interesting too.